Welcome, everybody. Again, my name is Brian Behringer, one of the coordinators and facilitators for the Memphis Scipreneur Challenge for 2021. I really appreciate everyone coming tonight and working with us through this virtualized environment to present 6 great pieces of technology from a variety of different organizations in town and a dedicated group of individuals who've been working on them for the last 9 weeks to present to you tonight, these great technologies. So hope y'all are in for a good show. We got a lot of great things to discuss and a lot of great presentations. So without further ado, I'm going to turn it over to Ryan and Erica, the coordinators for the Memphis Scipreneur Challenge, to start us off.
All right, thanks, Brian. First, I would like to welcome everyone to the 5th annual Memphis Scipreneur Challenge final pitch event and the first ever virtual rendition of this event. For those who don't know me, my name is Ryan Hughes, and I am the director of this year's Memphis Scipreneur. And so before we get started, I just want to briefly say a few words about this year's program. As everyone knows, this past year has been challenging, and Scipreneur was not immune to those challenges. I mean, it can be tough putting together a program and to get buy-in from participants when so much of entrepreneurship is the networking and the face-to-face interactions. But despite all the difficulties, I'm really proud of everyone who participated this year. Those who stuck with us throughout the entire program really showed tremendous resolve and enthusiasm. And I'm really excited that they have the opportunity to showcase tonight all the hard work and effort they've put into their companies these past 9 weeks. So before we kick this off, there are a few people I need to thank. First, Erica Dillard. She has been tremendous in supporting the Scipreneur program and helping me coordinate everything. I'm really not sure where I'd be without her help, so thank you. Then there's Brian and Shelby from CBU, who basically have made this entire virtual program possible. They have worked tirelessly putting together the curriculum, mentoring every individual, and leading the classes week in and week out. Their involvement has truly elevated this year's program, so thank you. And a special thanks to all the mentors and the entrepreneurs in the Memphis ecosystem who have given their time and expertise throughout the program, including our esteemed judges, who you'll meet shortly. And finally, a big thank you to all of our sponsors, the Memphis institutions, St. Jude, UT, CBU, and U of M, and most importantly, Epicenter and BioTN, because without their continued support, none of this would be possible. So thank you. And with that, I will hand it off to Erica Dillard, who will say a few words.
Awesome. Thanks, Ryan. I am going to need to share my screen briefly, if I could.
Yep, hang on one second. Go ahead.
Awesome. Oh, am I closed? Okay, now I see it. Sorry. There we go. You guys see my screen? Awesome. Perfect. So as Ryan said, I am been helping out with Memphis Scipreneur this year, and it's been a pleasure getting to know these teams. I am also the president of the Academic Alliance chapter here in Memphis. So I just wanted to give, for those of you who have never tuned into any of our activities before in the past, just wanted to give you all a brief introduction to what we're about. So our mission through Biotin Academic Alliance is to encourage Tennessee's economic and workforce development in the life science and the biotech. And we do this by providing graduate students and postdoc fellows an avenue for engagement with Biotin's statewide industry through events and programs that are aimed at several key areas. And those areas are networking, career development, public policy advocacy, outreach and mentorship. And as you will see tonight with the Memphis Scipreneur Challenge, one of our big areas of focus are entrepreneurship. So just a little brief update about what we're doing in the upcoming months. We are on Thursday, April 22nd at 5:30 to 6:30, we'll be hosting our second resume workshop. And the first one was done last year, but this will be the first one that we're doing as a joint effort with the other Academic Alliance chapters. So last year, we were able to provide key pointers from industry and academic institutional hiring reps on just how to make your resume ready so that job recruiters and interviewers will be kicking down your door for an interview. Also, as a perk of membership, which is free, we are providing resume review for those who would like a more in-depth assessment of their resumes. We've been collecting resumes over the past 2 weeks to provide time for review prior to the resume workshop. So if any of you are still interested in submitting your resumes and were unable to do this before the deadline, just shoot us an email at the email that you see here, jobfair@biotin.org. And we can see if we can work your resume review in with our reviewers, but please reach out tonight if you're interested in this. Our next event is our job fair. Our resume workshop leads up to our second virtual job fair, which was actually spawned through the Memphis chapter last year in August. So this year, we're excited to be doing this as a joint effort with the other chapters in the state. We had a wonderful turnout last year with over 70 participants from the Memphis area who were able to submit their resumes and were able to receive one-on-one interaction with hiring reps from the top companies in the state. As a result of this being a joint effort this year, we will have the opportunity to reach out to much more students and postdocs across the state and provide access to more companies as well. So you don't want to miss out on this opportunity, guys. Please visit our website at biotin.org for more information on how you can register for the job fair as a job seeker, or if you represent a life science or biotech organization in the state, whether it's academic or industry, with available positions that are ready to fill, please shoot us an email at jobfair@biotin.org for more information and details. And lastly, we have, let's see, there we go. Lastly, for our monthly networking event, which most of you have been able to attend in the past, our Beers and Careers will be coming up in July, on July 8th, 2021. So please join us to hear from the Patents to Products Fellows at the University of Memphis, who will discuss how they are using IP that's been created at U of M to spin out tech companies. Our own Ryan Hughes can attest that our competition tonight really gives a great introduction to what we're doing through the P2P program. And more details on registration for this event will be provided on our website and by emails to our members later on in June. So with that being said, just as a reminder, membership through Biotin is free. So you can visit our website for more information and to register for your local chapter. We're excited for tonight, and we're excited for the upcoming events through our Academic Alliance. So with that, I'm going to swing it back over to Brian to discuss how our program will be going this evening. So he'll talk to us a little bit more about how we'll do our program tonight. Thank you.
Great. Thanks, Erica. Again, my name is Brian Behringer. I'm the Director of Entrepreneurship at Christian Brothers University. I have my my right-hand person, Shelby Paranich, who's the Associate Director of the center as well. And we've been it's just been a pleasure for us to work with all the individuals on these projects, on these technologies throughout these 9 weeks. It's been very, very rewarding, hopefully for everyone. What we do at CBU is trying to push entrepreneurship out to as many people as we can through education and community outreach. And it's just been a real pleasure to work with Erica and Ryan and the rest of the teams from BioTN to as well as the Memphis Scipreneur Challenge. So what we're going to do tonight is we're going to walk through each one of these presentations. There's 6 in total. We have a panel of 4 judges, which I'll introduce here in just a second. Each team will have 7 minutes to present with 7 minutes of question and answers from the panelists of judges after each presentation. The panelists can ask them any question that they choose, and we'll just see how well they respond, combined with the responses from the question and answer period, as well as the pitch presentation, taking into account the quality of the presentation, the innovation of the intellectual property, and a variety of different things that would prove to a panelist that the team understands where they need to go to put something into the marketplace with their technologies. So we'll go through that tonight, one after the other. The teams have been selected in kind of a random fashion, so we'll go through those. I'll be calling out team numbers, and it might not go in order. That's because we did choose those in random fashion. We'll get started with that in just a moment. After that, we will conclude the presentations. The panelists will deliberate with a few of the coordinators that you've heard so far. They'll pick the winners, and then we'll come back. We'll hear from Abby from BioTN, and then we'll announce the winners and conclude the session for tonight. We're going to attempt to do something during the time the judges are deliberating. It's Zoom and it's all of its greatness. We're going to break y'all up into individual break rooms, or try to do so. You'll be paired with random other individuals. You can chit chat for 10 minutes. You can take a bio break. You can get another glass of wine or cocktail of choice, what have you. We're all at home probably. So please enjoy yourself while we're doing this, because typically when we're all together, we also are enjoying ourselves while we're watching these pitches. So please feel free. You have my permission to go into your own areas of cocktails and wines and everything else and help yourself. So without further ado, I'd like to announce our fantastic panelists of judges. These are individuals that have been in the entrepreneurial ecosystem for a number of years. A couple of these members have actually also been involved in the Memphis Scipreneur Challenge in the past. They've been mentors several times over. I've had the pleasure of sitting in front of these cohorts before with both of these individuals that I'm going to announce first. And Shelby and I are pretty well versed in what their backgrounds are, because we worked with them also at Startco and a variety of other things. So first, we have Isaac Rodriguez, Dr. Isaac Rodriguez. He's going to be heading up our biomed side from a co-founder perspective, along with another one of our colleagues. So Isaac formed Sweet Bio with his amazing sister. They're the power duo in their teams and their families and everything. They've worked extremely hard over the last half dozen years to bring Sweet Bio to market. They've gone through the entire 510K process and done it successfully, and they're working through that. So everybody, just do a good clap for Isaac Rodriguez for our panel tonight. Great. Love the golf claps. Everybody's on mute. It's perfect. See all sorts of emojis popping up, so everyone does know how to use Zoom after a year of using Zoom and WebEx. That's great. So next on the list, we have Josh Herwick, another colleague of ours. Actually, he and I were in the Startup Accelerator at Startco at the same time with his co-founder, Ezra Rohn, another biomedical product. Both Ezra and Josh are both biomedical engineers coming out of the University of Memphis and launched their own business a few years ago called Somovac, and it's doing great things. Josh is also, one of those people that have mentored people in this program before, and he's just a very knowledgeable person about the biomedical device side of things. So another round of applause for Josh. Great. Now I see even more emojis coming up. That's even better. Next we have Lisa Jennings. She's a global chief scientific officer for MLM Medical Labs here in Memphis, Tennessee. She's had a just historic career around the biomedical space. Definitely a go-to person when it comes to understanding the scientific space and intellectual property and tech transfer process. She's just a great resource, and we're really, really happy to have her along the ride for tonight as well. So another round of applause for Lisa Jennings. Okay, all right. And last, and certainly not least, we have Jan Boone. Jan is the founding partner of Innova Memphis. He's also been a guest speaker during our cohort this year to talk about venture capital funding. He's certainly one of the biggest early stage investors that I know around here and anywhere. He's certainly kind of paving the path in that regard, and has been doing so for a long period of time here in Memphis with Innova. So he's a great resource from a funding perspective. He's seen thousands and thousands of pitches over the years. He's probably doubled me by now, for sure. And he's just got a wealth of knowledge in the biomedical space. He helped set up the UMRF projects, patents of projects, and that foundation fund that he manages for that as well. So great resource with Jan Boone. Everybody give one more round of applause for Jan. All right.
Hey, Brian, before we get started, Kayla also, make sure to introduce Kayla too.
Oh, okay. Kayla Rodriguez, the other power half of the Isaac Rodriguez duo, amazing person. I've had the pleasure of knowing her as long as I've known Isaac. I've often worked with her, both just talking about Sweet Bio and other businesses and where they're going and everything else. She's a fantastic resource. You can't beat this power duo that have formed Sweet Bio over the years, and they're showing great success and their strength as a team and a unit. So one more round of applause for Kayla as well. Okay, so we're gonna have one of the two Rodriguez's at least be a panelist, if not both. So I might have gotten that a little incorrect. So don't worry about it. We'll move forward. So without further ado, I'd like to go ahead and get started with the presentations. We're going to start off with Team 6, Rohan, and he has the ability to start sharing. When you get your slides up and ready to go, we'll start the timer. Again, each team has 7 minutes, and we are going to send out a virtual hook to pull you away right after that. So let's hit that mark. Panelists, you'll have 7 minutes to ask questions after that. Okay, so I'm going to give you the countdown. 3, 2, 1.
Hello. Hello, everybody. Good evening. It's my pleasure to introduce you all to our solution, the true tummy time solution, which is a solution that to a problem that faces thousands, if not millions, of babies, like young Timmy here. The problem is this lack of prone play or tummy time. This lack of prone play or tummy time in infants can negatively impact their motor development and can cause and worsen flat spots on their head. Studies show that over half of infants receive less than their recommended 30 minutes of tummy time per day, and over a third are intolerant of this position. The American Academy of Pediatrics recommends 30 minutes of tummy time per day in order to avoid these developmental issues, which can cause to need solutions such as the headgear you can see on the cute baby on the right. The market is a surprisingly large market of babies that this problem affects. Over 56% of twins, coming to a total of 67,000 twins each year, are affected by this problem. And even though the fraction of single babies that are affected is much smaller, it still comes out to a total of 4.8 million babies each year. Treatment for such a problem costs anywhere between $2,000 to $4,000, which can be a significant burden on parents during this first year. You can see the repercussions of this flat head in the pictures on the right, where these young infants develop these flat spots on their heads, which can take a long time and a lot of money to fix. Our solution to this problem that affects so many babies is a true tummy time wedge solution. It's a unique system of support and engagement for babies in this developmental period. Our unique value of our product is that it consists of 2 form-fitting wedges. These wedges have a molded design that shifts the baby's weight to their hips. This avoids pinch points under the armpits that are found in most other products. It also consists of different sizes to help grow with the baby. In addition, another key point of our system is the addition of a play wedge, which includes multi-sensory toys and a baby-safe mirror, which encourage babies to stay in the prone position and make the development in that position a lot more easier and comfortable for these young infants. To compare our product to the others in the market, it stands out in a number of ways, overwhelmingly. Some of the key points I want to draw your attention to is the full body support in our product that is absent in any competing product. In addition, it's the only product on the market that is backed and supported by research, which gives it a huge edge over the competitors. In addition, it has a number of other advantages, as you can see, compared to the other products on the market. To take this product to market, we have a number of different ways we can target. We can either target directly to parents in the form of direct marketing, baby gifts, or we can target to institutions. And our target to institutions is backed by our research background, which other products do not have. And thus we can easily target daycare centers, pediatric institutions, and even market our product through parenting groups. Our business model at this early stage will be direct to consumer, driven by web sales, personal recommendations, and social media, and other forms of direct marketing. In the future, however, as we scale up, we have an opportunity to take our product either directly and put it in stores or partner with manufacturers to add it alongside their lines of products. The total baby market is huge, and we can easily tap into a small fraction of this large market. The average household spends over $13,000 a year on baby-related products alone, not counting medication and other medical-related expenses. The price point of our product is slightly higher than competitors at 80 to 100, but we believe that the advantages that I've shown before more than make up for the small increase in price. The cost and labor of making these products, even including overhead, is much lower, and we can make our income directly from that difference between the price point and cost, which allows us to directly start making sales immediately. In the future, as we sell more and more of these products, we can expect to scale up pretty easily and considerably, and hopefully continue making significant margins. So far, we have taken this product from a prototype, designed and developed a prototype, gotten funding from various sources on this prototype, moved into doing studies where we use user feedback to help improve the product. We're now in the process of reviewing these information that we've gotten from our customers and resolving and improving our prototypes in order that so that we can sell our product. Our projected future timeline at Ask involves first 2 rounds of milestones. Our first milestone involves a beta run of our current product. And the main focus in this first milestone will be to focus on R&D to improve and iterate on the product in order to improve the features and the user feedback. You can see below some of the details of the prototype and what the revisions are starting to look like. The next milestone will involve a revised product run based on all this initial feedback. But the focus in the next stage will be not to focus on R&D anymore, but instead to focus on marketing, website, advertising, sales to help drive consumers to our product. Our team consists of Dr. Ann Zachary, who's the inventor and the project lead. She has over 30 years of expertise dealing in this area. We also have me, Rowan Isaac, who's helped develop this product, as well as Shelby, who's helped advise and move this product along. With that, I just want to say thank you. And if you're interested in learning more about our solution to this very interesting and widespread problem, please reach out to us. Our goal is to make tummy time safer and better for parents and infants alike. Thank you very much.
Thank you. Nice job. Nice job. Okay, so we're going to open it up to the panelists, and if everybody else can stay muted, but the 4 panelists that we have, Kayla, Josh, Lisa, and Jan, if you want to speak up and ask a question, now's the time to do so. We have 7 minutes.
Yeah. Hi, this is Lisa Jennings. I'm just interested a little bit in the pinch point. You know, that sounds like something that parents would want to address and, you know, might be, you know, somewhat emotive. Can you talk about the advantages of your product in terms of pinch point and what is that exactly?
Yeah, absolutely. So if you see our product in either the wedge form with the baby over here, you notice that the under the armpits right now, there's no pressure directly on those armpits. Our competing products are generally involve either rolls that are either completely cylindrical or have some sort of C shape that only supports the region under the armpits. And that puts a lot of pressure on the sensitive armpit area, which can be very painful and make tummy time not very pleasant for babies. But seeing that they're simpler, smaller, they tend to be the most widespread product that's currently used on the market.
Mm-hmm. So what's how do you clean that? I mean, parents are going to be interested, particularly in view of the fact that you know, we're all dealing with you know, not so much COVID. I'm not concerned about that, but just it looks like it's somewhat convoluted, or you know, there's a lot of movement in the surface there. What do you have any recommendations, or is it easy to maintain?
Yes, so the cover is removable. The inside is purely upholstery foam, and the covers are removable and completely washable. And so cleaning the products should be very straightforward. Yeah, okay. I don't have a lot to add there, but I think most of the products out there are fairly good in that regard in terms of you can mostly just throw them in the washer and they'll be just fine. The cover being removable is a little advantage, just so you don't have to stuff the entire thing in an oversized washer. You can just take the cover off. The toys can be sanitized, but yeah, I think the same goes for just general infant toys. Did that answer your question?
Yes, thank you.
Thank you.
I'm happy to jump in.
I had a question about the IP. I mean, you didn't say much about whether there's patents for this or whether any design proprietary stuff. I mean, it seems like a lot of development work, but once you see it, it's probably fairly easy to copy, right? You take the cover off and scan it and print a foamy thing. So what's stopping anyone from taking it?
So I think you raise a very good and valid point, is that there's not a lot we can do to stop the spread of people copying this, but what we do offer, the advantage that we can back is our
Looks like we might. Rowan, you there?
So if we have a knockoff, for example, they may look and seem pretty similar, and visually you might be able to say, oh, this should work pretty well, but we can actually verify that we've taken these products, we've done trials, and here are the results. But you're right, it's not a perfect system. We do base it on our brand versus actually having a stamp of weight that we can enforce someone making a similar shape. Did that sort of answer your question?
Yeah, you broke up sort of halfway through, but I think we all got the gist.
Oh, I'm sorry.
It's like you have data and backup analysis, but you don't have a patent.
Yeah, I think the advantage we're going for is that it looks, and I'm curious if the technical lead and inventor has anything to add on this. Dr. Zachary, I believe she's here. But I think, go ahead, Dr. Zachary.
It does have a patent pending. Its application has been submitted for the patent.
Awesome.
Yeah, yeah, sure. Sorry, I keep jumping in if you see me popping up, but I'm a mom of two. I've got a 3.5-year-old and a 1-year-old, and I've had 2 kids that scream every time during tummy time. I am quite scarred from this experience. So I would pay for something that would, and I think that your price point does make sense. It's in the market of what typically I'd say like these luxury baby products are. A couple of things, that I would think through on your go-to-market strategy. A first mover advantage does play well in this space. I do think in most industries, first mover is kind of a, not the strongest position, but in the baby space, who gets there first and who looks the best. Branding is absolutely everything. So my thoughts when I saw your slides are, how can you invest in branding and look like something like a Snuggle Me Organic? They excel at that, as well as how do you get on with the top blogs or the top books like Moms on Call. Like there are some really leading organizations. You're smiling because I'm sure you know them, but there's some leading organizations out there that allow this first mover advantage, and I think that can address a little bit of the copying if you continue to put out new things. But yeah, I was also very curious on, as a mom, I also like research, but what does your research show? Is it the pinch point, or what exactly is the data point, that one line that you would put for that parent?
If I was to put one line for the parents, it's comfortable and they enjoy it. They enjoy it more, so they're willing to spend more time on it. So the fact that we have both engagement and it's comfortable, the pinch points are a concern, but it's a concern for both the baby and the parent, right? So if the baby's getting the pinch points, it's making a lot of noise, it's unhappy, it doesn't like the position. But at the same time, the parents are also unhappy because the baby's unhappy. So I think the comfort level is really what we're going for. It's comfortable, they're enjoying it. It eases them into that development of those muscles in the back, neck, and the shoulder that takes and makes the tummy time uncomfortable.
So
I'm also a former Target. So if you guys keep this going, let me know, and I'm happy to see if I can get you connected with Target. So
Okay.
Rohan, I had just one more question. It seemed like, if your cost to produce it is between 50 and 70 and your price may hover between 80 and 100, is it possible to do better on the cost of production? Because, I'm not too impressed by the margins here.
Yeah, so that's an extremely valid point. And the margins are very much right now, early stage. All the stuff is being done by hand. The prototypes are being sort of carved out. And so I think any sort of scale, it really lends itself towards, so it's not been scaled up yet. And so the margins are kind of slow. And I wanted to keep a sense of that there are a lot of things that you can bulk up, right? So even if you just buy upholstery foam, if you buy 10 of it versus 100, you just make savings right there. You don't even have to scale it up. Even if you're still doing it by hand, you're still saving money that way. But right now we're just small prototypes getting R&D, so we have small margins, but hopefully as it scales up, those margins will continue to grow between the cost and the price of the good.
Yeah, I mean, for this type of consumer product, you want to shoot for that the sales price and the cost should be like a 1 to 3. So if you sell for 80, you should be able to make this for 25-ish.
Yeah, and I think if you look at just actually the cost of the materials, I think that's very reasonable in terms of it's literally just foam and cloth. The actual materials don't cost much, but when you're doing it in small amounts and you have workers that you're having to pay to cut that stuff up, it adds up at our early stage. But yeah, I think it's pretty easy to move out of. But yeah, thank you both for that feedback. I think that's extremely helpful.
Okay, well, thank you very much, Team 6. That is the end of the allotted time for questions and answers. We're going to try to get to at least 3 panelist questions for each round. I really appreciate those questions. Thank you, Rohan and Team 6. So next up is Team 2. So Team 2, when you are ready to make your presentation, you can share your slides, and when I see them up, I will start the time clock.
Okay, I hope everyone can see the slides in full screen.
Yep, we sure can.
All right, so.
Okay, let me start the clock, and you're good to go.
All right. Good evening, everyone. My name is Pankaj, and I'm excited to share our discovery about precision therapy for leukemia. Leukemia is one of the major types of cancer all over the world. In the United States, more than 60,000 people will be diagnosed with leukemia in 2021, out of which more than 23,000 people will die because of leukemia. If you look at leukemia death rates in the United States, it is pretty much concentrated in the central region around Memphis, where most of the deaths have been seen. And the age group which is highly affected by leukemia is unfortunately children. I still remember my first day at St. Jude when I joined St. Jude and I was going down to take my food. There was one little girl just like this one standing in front of me, pushing her chemotherapy IV drip along with her as she was waiting to get the food. When I saw this, it literally broke my heart. And I started reading more and more about what goes wrong in case of leukemia. In the leukemia, there is one particular pathway called as JAK-STAT pathway is hyperactivated because of either mutation in JAK2 protein or overexpression of JAK2 protein. And because of activation of this pathway, it actually starts a domino effect where the leukemic cells, they proliferate more and more, and thus leading to leukemia. All the current chemotherapy drugs which are available right now, they are not specific for JAK2. And since they are not specific for JAK2, you have to use high amount of drug to get the similar side effects, which leads to more side effects of chemotherapy. Unfortunately, there are no orally bioavailable drug for treating leukemia. So patients have to rely on chemotherapy, IV drips, as well as hospital visits, and which eventually leads to poor outcome of leukemia. So all these current drugs, how they actually work, they are actually inhibitors of JAK2. So one molecule of this drug, it goes and binds with JAK2 and thus inhibiting the JAK2 protein. Remember, this is a stoichiometric process. So only one molecule of inhibitor is going to block only one molecule of JAK2. So you need more amount of inhibitor to block more amount of JAK2. At the same time, cells contain an inherent natural ability to get rid of all the unwanted or misfolded protein by a worker called as E3 ligase. But now, how this E3 ligase will know that JAK2 protein is actually harmful and you need to degrade that? This is where our invention comes into picture, which is called as PROTAC, or proteolysis targeting chimera. This small molecule at one end very specifically binds with JAK2 protein and literally brings it closer to this degrading protein. By this way, it not only blocks the JAK2 protein, but also leads to complete degradation of JAK2. The most interesting part, this PROTAC molecule, once it degrades this JAK2 molecule, it is again recycled back so that it can bind to the new JAK2 protein and thus degrading it. So this particular PROTAC is highly specific for JAK2. And since it's more than 100,000-fold efficient than the current inhibitors of JAK2, you have to use very low amount of it. And most interesting part of it, the PROTAC works in a catalytic manner, which means that single PROTAC can degrade multiple JAK2 proteins. And the molecule of PROTAC can be easily modified so that it can become orally bioavailable. So we have compared our PROTAC molecule with the all existing blockers of JAK2 as well as the antibody therapy. Our PROTAC has many advantages over the current therapy. We also compared our PROTAC with the similar molecule developed by these three companies. However, our PROTAC actually shows more advantageous features in terms of ease of developing, catalytic mode of action, a very small or picomolar activity, and specific degradation of JAK2 protein. In terms of our company, PROTACure, we have developed it, we are going to develop it into 3 different phases. In the phase 1, we will be requesting for the exclusive IP licensing from the St. Jude Children's Research Hospital in return for the royalties. We will be establishing our company at Memphis, which is in the center of all these leukemic cases which has been reported. We will start with the preclinical studies for the safety and efficacy of our PROTAC product. And while we are doing the preclinical studies, we will be spreading awareness about the PROTAC and its efficacy in various national and international conferences like ASH, American Cancer Society, Leukemia and Lymphoma Society, as well as AACR. In the second phase of our company, we will be giving the non-exclusive IP rights to one of the major pharma companies like Pfizer, Johnson & Johnson, Novartis, and AbbVie, which will then lead to the GMP manufacturing of PROTAC at a small scale. This particular GMP manufactured PROTAC will then be used for the clinical trial. For this clinical trial, we'll be recruiting multiple hospitals, including MD Anderson, St. Jude Children's Research Hospital, as well as Johns Hopkins. All the data which will be generated from this particular clinical trial, it will help us designing new PROTAC targets for various oncoproteins as well as neurological proteins which are there in the disorders. So for this one, the progress which we have made so far is we have already conducted the research and discovered JAK2 PROTAC. And this is an interesting result which you should be noticing, that these are the mice which has very high leukemic burden, which you can see with the bioluminescence by the green color. If you treat these mice with JAK2 inhibitor, which is ruxolitinib, you can see that the leukemic load still remains the same without reduction. But if you treat them with the PROTAC, you can see that these mice are now leukemia free. This was very encouraging, and we have presented our data at Cold Spring Harbor Laboratory as well as Transcription Therapy Symposium. In the second half of 2020, we filed for the patent, and we have got the patent with this particular patent number. We have submitted the research paper, and early in the 2021, I'm very happy to share with you guys that our paper has been accepted in one of the reputed leukemia journal called as Blood. We have also initiated the exclusive licensing talks with St. Jude's Children's Research Hospital. Our executive team has been formed, business plan has been developed, and we have also pitched this idea at MSC 2021. So for the first phase of our company, that is foundation and validation, we will be asking for $390,000, of which $90,000 will be used for obtaining exclusive license from St. Jude Children's Research Hospital, and $300,000 will be used for the preclinical studies. This is our team. Dr. J.K. Mean is our mentor. He has more than 30 years of experience in chemical as well as pharma industry. Myself, I'm a stem cell biologist as well as molecular biologist, and I have 15 years of experience in this field. And our team is joined by young aspiring entrepreneur from University of Tennessee, Kathleen O'Brien. With that, I would like to thank you all for your time, and a special thanks goes to all the MSc mentors, Brian, Ryan, Shelby, and Erica. Thank you so much.
Great job, team. Great job. Okay, let's turn it over to the panelists.
Okay. I wanted to know how did you come up with the money that it's going to take to get through kind of your first phase? You had about 390,000. I saw the 90,000, but is 300,000 enough to get through clinical studies and that type of thing?
Yes, at least for the initial phases, for the preclinical trials, yes, it would be sufficient. But for the clinical trials, no, it is not.
So how far will that actually take you?
That's a great question. So I think this will help you understanding it further. So basically, from the seed funding which we'll be getting, we'll be doing the preclinical studies, and then we'll be sub-licensing it to the major pharma industry. This pharma industry will then carry it forward through the clinical trials, because we being the startup company, clinical trial requires anything upwards of $21 million. So we probably will not be able to raise that much of money. So we'll be sub-licensing, and in terms of that, we have did our maths where we have seen 10 different pharma companies and their drug manufacturing cost as well as their revenue in 4 years for any of those 10 drugs. So the median revenue for any cancer drug is around $1,500 million. So these companies will be paying royalties back to us, which is usually in different forms. Like they will be paying royalties in terms of the patent filing money, as well as the upfront fees, as well as the milestone payments. So even if you consider 7% of the net sales for the cancer drug, we will be getting in return around $100 million total, which will be more than sufficient for our next R&D and designing of new products for different targets. I hope that answers your question.
Yeah,
Sorry.
no, absolutely.
I'll ask a question if nobody else is saying anything. What about the patents here? Are they in progress? Are they issued? Where is it with the patent office?
We have already filed it. We have got the patent number, and I think it has been finalized. I'm not exactly sure.
Right, but it sounds
Yes.
like it's not issued yet.
It's not an issue, yes.
Yeah, it's not issued yet. Thank you, Jackie.
So just a couple of questions. So you do have an agreement with, I assume the owner of the IP is actually St. Jude.
Yes.
Since you're employed by them, are they in agreement to actually license you this technology? Has that been established, or is there any bumps in the road in terms of being able to license that?
That's a great question, Lisa. Yes, we have already contacted the office, and we are already in terms with them for getting this exclusive license with them.
And how did you determine the specificity of your kind of target or probe, if you will, to JAK? Because that's in the immuno-oncology area, that's very much a focus because of just potential off-target effects and such. Have you extensively looked at the specificity, or is that still left to do?
That's a great question, Lisa. And thankfully, I have this figure to show it to you. Basically, any other product, if you use JAK2 specific products or JAK2 specific products which have been available out there, they are not specific. They leads to the degradation of another subsidiary protein called as GSPT1. But our product, as you can see here, it specifically leads to degradation of JAK2 with an increasing amount, whereas it does not hamper any other protein, especially GSPT1. So yes, we have seen these studies in case of 10 different cell lines, not only one, 10 different leukemic cell lines. And we have seen that our PROTAC specifically degrades JAK2 protein.
Okay, I think there's even more sensitive screenings for that. So, you might consider that because, the gels are certainly give you good evidence that you're heading in the right direction in terms of specificity. But I know from other experience with other pharma targets, a little more high sensitivity might be in order as you move forward in your development of your drug.
Yes, that's correct.
I would agree. And I think jumping back to Josh's question about the 300,000, does that just cover, and maybe I missed it, but it's always going to take twice as long and 2 or 3 times as much. And I think it'd be helpful to even ask Pfizer what they would want to see, to Lisa's point, or Dr. Jennings' point of, what kind of testing, because you have a chance right now to go collect data that they're looking for, but also create data they've never seen before. And so allowing, raising another $100,000 or $200,000 could give you that freedom to do some tests that aren't just regimented and that you're trying to, get to your final answer of. So I'm curious, of the 300, is that based on exactly what you need, or do you have some flexibility in there to kind of go off path and create some new data points?
That's a great suggestion, Kayla. And yes, we do have flexibility regarding this. So the 300,000 figure we have came up across through different costs we are going to require, like protocol writing, animals required for the study, as well as manufacturing of this PROTAC, and then establishing the study and doing it in a statistically significant manner. But yes, more money will definitely help us using different parameters and different tests. Yes.
We have time for one more question.
This might be a big question, but so PROTAC is kind of generally the technology, and JAK2 is kind of the first target, for leukemia, obviously. What other things are you potentially considering, or have you thought of that yet in kind of the phase 4 way down the road?
That's an excellent question again. So basically, if you look at various oncoprotein in these particular disorders, as well as, so there is a whole lot of array where one particular protein is either overexpressed, that is the oncoprotein, or different proteins in these particular disorders. Also, if you remember, there is a whole array of neurological disorders which specifically occur because of accumulation or aggregation of protein, like Alzheimer's disease, Parkinson's disease, and CJD. So since these are all protein-based disorders, you can specifically design a PROTACs which can bind with them, and thus leading to the degradation of these proteins. So yes, we have future plans for designing new PROTACs for all these proteins.
Thank you.
Great. All right.
Just one more question. What about the JAK2 mutants? Do you have any data on that as far as your compound? Is there any difference in JAK2 mutants versus JAK2?
So this particular PROTAC actually binds at the active site of JAK2, and most of the mutations occur at the other positions other than the active site. So basically, our PROTAC is equally effective in case of mutants as well as the normal JAK2 or overexpressed one.
Thanks.
Yeah.
All right. Great job, Team 2.
Thank you. Thank you all.
Okay, so Team 3, go ahead and cue your slides up for us.
All right. All right, does everything look good?
We're seeing your preview monitor. You might want to switch to your presentation monitor. You can do it. Yeah. There we go. All right, Brad, count to 3. 1, 2, 3.
All right, my name is Brad Bomar, and our company is called Helisan Innovations. So this is Jim. Jim has diabetes, and he just had his right leg amputated. And you may be wondering, how does this happen? So let me show you a bit more detail. This is a diabetic foot ulcer. This is how it starts. So to summarize, diabetes can lead to poor circulation in your extremities and neuropathy, which can lead to a foot ulcer showing up without a patient knowing or without taking their sock off to find out. 50% of these diabetic foot ulcers become infected, and 20% end up having to have an amputation. So if you just look at the US market, 34 million people today have diabetes, and 2.25 million have foot ulcers. And every year, 73,000 patients have to get some form of a lower extremity amputation. Here's our solution. We call it Helisan. And this is just a CAD model that we made, kind of showing the details of the product. So Helisan is made up of chitosan, and chitosan comes from chitin, which is the exoskeleton of shellfish. And what's really cool about this biomaterial is it has innate antimicrobial, antibacterial, hemostatic properties. And Dr. Jennings has done research and found out that if you combine polyethylene glycol with it, it actually attracts hydrophobic loadable therapeutics such as bupivacaine or marcaine. And we also have skin attachments on our product that hold the wound contacting layer of chitosan. So what's the value here? So antimicrobial, antibacterial properties prevent infection. Skin attachments kind of allow you to have no extra cost to your hospital or your clinic, because if you have a product that you put over a wound and you need something else to attach it, that's just adding cost and burdening the US with more healthcare costs. So hemostatic stops the bleeding, and loadable therapeutics can be chosen by the provider, according to Dr. Jennings, at the point of care. So we all know the wound care market is huge, right? It's $20 billion a year. And so with that, there's a lot of competition. And a lot of people have silver dressings, Manuka honey dressings, but we say, why add something to your product when you could just choose a biomaterial that has the innate properties itself and lower the cost of your goods? So here's our strategy. We looked at the wound care market and we said, who's taking care of these patients? Podiatrists are taking care of these patients, is what we found out. In the US alone, there's 15,000 podiatrists. And just to remind you, there's 2.25 million patients every year with diabetic foot ulcers. Our sales channel, what we're thinking is it may be easiest to use independent distributors that are in the foot and ankle orthopedic market that have relationships with podiatrists, and try to get in on some of these debridement surgeries that occur and get in the global reimbursement period of that surgery. That may be the easiest avenue that we're thinking. So here's our business model. $40 average sales price, $15 cost of goods, and a 62.5% profit margin of those rough numbers. And this is just from our research, from what we're trying to find out and get competitive pricing and look at the market and really assess who can make this product and who can sterilize it and package it and give it in a form that can be delivered to the podiatrists. All right, so let's look at revenue. 2.25 million diabetic foot ulcer patients. And we did some market research. We found out that after a debridement surgery or a amputation surgery, typically what the podiatrist tells the patient is to change their dressing twice a day for 14 days after surgery. And we said, wow, that's a lot of dressings. And we're being conservative here and realistic and transparent. And so if you say 3 times on this middle circle, use a $40 Helisan dressing, that's $270 million in revenue. If we capture 0.1% of this market, that's $270,000. So our traction currently to date has been awarded grants with Dr. Jennings. She has done a great job and published proven research on this biomaterial. She's done characterization studies, looking at all the safety, biocompatible properties of this material, as well as Dr. Haggard and Dr. Baumgartner in the biomedical engineering department at University of Memphis. And the really nice thing that we found was they have granted patents. There are 2 US active patents. One of them is on chitosan polyethylene glycol and a loadable agent, and the other is the film and sponge material makeups. Here's our ask, $62,000. And you may say that's kind of low. And we agree. We think a low cost investment that's low risk to get a feasibility to make some prototypes and to spend it on, if you look at the top circle, first on prototypes, then on verification and validation testing for our 510K, then on the 510K application, then in parallel path, look at our customers and ask if they'll make commitments, and then file IP provisional applications and have office space for us to work. Here's our team, as well as our board below. Thank you. And we'll take any questions.
Thanks, Brad. Great job. Since you called someone particularly out, I'm going to go directly to Kayla Rodriguez Graff from Sweet Bio.
John, let's go. No, great job. I do have a couple of questions.
Sure.
So the first one is what are you expecting from reimbursement? And then just let me set the stage for a second. You know, if you're looking at physician offices and debridement charges and office visits, there's only certain things that you can do, and you're in a pretty tight window of costing, and they aren't using dressings, which this would fall more towards that end. So I'm curious, when you're looking at your total addressable market, especially in the podiatry space, what conversations are you having on reimbursement? Yeah, I'll pause there.
That's a great question. So if you look at my previous slide here on customer commitments, this $4,800 was going to dive into that and really dig deep and ask those hard questions to the podiatrist. So that's going to find out, well, is it debridement surgery that we should look at? Is that costing too squeezed by CMS and Medicare and Medicaid? Or should we go to the amputation surgeries? Should we go to, you know, different parts of your care? Like, this $4,800 is going to dive deep into that question and find out specifically where this product could fit best. But that's a really good point, though, because, you know, CMS is always squeezing on pricing. Like, and if you can find a lower cost of goods to lower your pricing, that's always better. And you can always beat the competition that way in a competitive market. But that's a great question. I don't know if that answered it, but.
Yeah, and I think if you can invest in that now, it could change the words that you use in your application. It can change your predicate. It can set you up for success if you start having those conversations earlier on.
Right.
And then my second, I would say, comment is, you know, it costs $3,500 just to submit an application to the FDA. So I would definitely, and they were probably going to ask for some chemical characterization data across 3 lots of time points. And so I definitely take a look at these numbers and make sure you're asking for, you know, what you need to be able to make that application a success.
You're actually describing the verification validation testing, this $28,000 here. So this is going to go as proof, and the data is going to be entered from this testing into the application. So that's a good point. I probably should go back and see if 28,000 is enough, though.
Yeah, I'd say
you go ahead.
I was going to say V&V testing, it's probably 20 times more than that, 10 to 20. Whoa. I don't know, by the time you get it done. So on that alone, budgeting for IP, you know, you got to think about countries, and once you have to actually submit the utility and all that. So
it's true.
I know there are 2 people on this call that could help you cost some of that out probably a little bit more accurately.
Oh yeah, actually, if you see my contact info, I'd love to connect with them. So if you could send them my way, and maybe I can get a little bit better idea about the countries that may be good if we're looking outside the U.S., the cost. So I've drafted patent applications for Smith & Nephew. Crossroads is pretty good. Chad Hollis does a lot of those, and he has a patent agent himself, and he's my boss. That's a good question, actually. And if you could send them my way, I'd appreciate it.
Yeah, I would tend to echo a little bit about the costs. We just submitted, in fact, a 510K today, and you know, there's a lot of cost. It's really unbelievable, even with a predicate, and you know, our particular material was an implantable surgical device, so you know, more concerns in terms of biocompatibility, you know, but it did have a drug on it, and so you know, it becomes a little more complex, but there are a lot of costs associated with that, but definitely a needed product. And you know, I think kind of your strategy is a good one, but I think it will take a little more money than you're asking on the front end. And you know, nothing's worse than to feel squeezed on the finances and then you know, necessarily having to go back and ask for more money. So probably a review of that budget might be a good idea.
Okay. Well, what do you guys, if I could ask, I guess, Kayla, Josh, and Lisa together, like, what do you think a better ask would be than 62,000? What do you think I really need?
I think one
good. Had to give a good guess.
Well, it may not be that the number is wrong, just that your endpoint is, you might want to adjust the endpoint. Obviously, it's a lot of faith for someone to give you enough money to really get through to a 510K submission, but you might think about proof points ahead of that. And that would be more in line with what you know, very early stage investor would give you at this point. I think there are a lot of other questions in terms of where the IP is at and that kind of thing, but
okay.
It's probably more than the minute we've got left.
Yeah. So Brad, the idea is for them to ask you questions, not you ask them.
That's all right.
That's all right. That's all right. We have time for at least one more question, Jan or Lisa or anybody.
Yeah, I think, I mean, in general, I mean, you have two issued patents. I kind of wonder how much they really cover this product. And whether you've looked at what else you might have to file, because the chitosan space in general, it's a pretty tricky IP landscape. So who have you talked to on the IP side to make sure that you actually have what you need to bring this product to market?
Sure. So,
and budget about 5 times what you're going to say for that.
Okay. So the 2 US patents that have been granted, the first one, I looked at the independent claims and they're actually pretty powerful. I was surprised that Dr. Haggard really did a great job getting it. It's chitosan with polyethylene glycol, and the language in the independent claim says a loadable agent. And I saw agent, I'm like, that is really broad, like that's really powerful. And the polyethylene glycol is to make the chitosan hydrophobic, so it attaches and loads and stays with the wound contacting layer. The second one, the independent claims are pretty powerful around just chitosan sponge and just chitosan film. And both of them have independent claims on the method of manufacturing as well. So I thought it was a great job that Dr. Jennings, Dr. Haggard, and I believe maybe Dr. Baumgartner helped with on getting those current 2 granted active patents. The third one that we're thinking of that we should probably file a provisional on pretty quickly is to add the skin attachment, maybe with a continuation if we can. I don't know if there's any, Dr. Jennings, maybe you can hop on and answer for me, but the skin attachment with this to really capture our commercialized product and prevent anyone from doing this would be the first thing I would file.
Yeah, I mean, a continuation, I guess it just depends, you know, if you've not had it in the parent application, you may have to go in for a new provisional, but it sounds like it's worthwhile moving forward on that.
Yeah.
Okay.
So, and I'll add to, since I was involved in this, that, so some of the different aspects can add a lot of value to this. So, it could expand the applications as well.
That's right, because there's a lot of other applications. We're very focused and driven and looking at one space and market, but Helisan, you can actually probably take this biomaterial to the hernia mesh market and general surgery, like military trauma wound applications, maybe even look at IP licensing deals with other companies like Somovac, to maybe put the Helisan onto the patient skin contacting layer, like behind the I guess the attachments on the Somovac device. Like we have a lot of ideas that maybe we could dive into that would require another race.
Yeah, we can dive into those another time when we don't have 3 more teams to get through. So Brad, thank you very much. You've done a fantastic job. The questions are great, responses are great, and we're going to move on to team 1. Good job, Brad. And
all right, thank you guys.
All right, team 1, start spooling up your slides and I'll start your timer when I see it in full screen.
I'm sharing my screen.
Yeah.
No. Can you see my screen?
We see it in presentation mode now. Great. Okay, I'm going to start the clock in 1, 2, 3.
Hello, everyone. I'm Panduran Kolikar from Team 1, representing our scientific business venture, Dogma Bio. It aims to provide smart software solutions to empower early detection of cancers. When it comes to the late stage of cancer, patients and families have to go through a lot of hardship. This family from Canada is spreading awareness by sharing its life-changing experience due to early diagnosis of their child. It has been shown that liquid biopsy methods are very useful in early diagnosis of cancer, which increases survival rate up to 90%. But imagine, in case of late diagnosis, the experience of this family would be devastating, as only 10% of the patients survive late diagnosis of cancer. Sorry, I got this thing. Yeah. So, different types of cancers in their various stages of growth release tumor DNA in blood. These blood samples are then subjected to DNA sequencing to detect specific mutations which help in decision of treatment. Thus, liquid biopsy can be used for early diagnosis and to monitor drug response during treatment of cancer. However, during early stages, the proportion of cancer cells in blood go down below 0.1%. Sequencing errors further adds to the complexity in detection of these low-frequency target mutations, hence making it a very challenging problem, like finding a needle in a haystack. This significantly limits the accuracy of liquid biopsy methods in early diagnosis. The standard analysis methods fail to distinguish such low-frequency target mutations from sequencing errors. To overcome this challenge, we developed a computational method for systematic separation of sequencing errors to such an extent that mutations with frequency as low as 0.05% can easily be detected. Our methods are thus helping researchers to solve this needle in a haystack problem. Our method has also been successfully applied to detect the drug-resistant mutations about 4 months earlier in leukemia patients. Such level of advances in early detection help clinicians to effectively devise new treatment for patients, giving them a hope for better survival and quality of life. Though the main beneficiaries of our solution will be cancer patients, we will be targeting major liquid biopsy companies like Grail and Guardant Health as our customers. Subsequently, we plan to serve hospitals and cancer research centers with various applications of liquid biopsy methods. In a nutshell, our methods offer 100x better resolution, 3x reduced cost, and faster turnaround time, which means a better quality of life for patients. Currently, Grail, Foundation Medicine, and Guardant Health are the major players in the liquid biopsy industry. However, in terms of separation of error rate, Dogma Bio shows 100x better performance. This further helps us to achieve a limit of detection as low as 0.01% compared to 0.1% by Grail and others. Furthermore, Dogma Bio does not require UMI-based redundant sequencing protocol. Thus, the cost of sequencing, data storage, and analysis time is much lower compared with other companies. We plan to utilize digital marketing tools such as websites, email marketing, and SEO techniques. We will leverage the scientific articles and white papers for B2B content marketing to major liquid biopsy companies. We plan to utilize social media platforms, blogs, and newsletters to create awareness among customers and the general public. As part of field marketing, we will participate in scientific conferences and sponsor webinars to share the applications and success stories of our products. This will definitely help us to generate leads and have face-to-face meetings with our potential customers. We plan to employ B2B business model with software as a service. Our software solutions will be deployed in a secure, scalable, and HIPAA-compliant cloud infrastructure. Our customers, such as liquid biopsy companies and hospitals, will upload sequencing data from a number of liquid biopsy samples using interactive interface or in an automated manner. Dogma Bio solution will then process the data and provide accurate reports to the customers in a timely manner. Currently, on an average, we plan to charge $10 per sample with an estimated processing cost of $4. Our pricing strategy is subject to change with respect to factors such as competitors, priority of the analysis, length and size of contract, and additional services. Currently, patients visit major hospitals and research centers for diagnosis and treatment. These organizations, in turn use the liquid biopsy services provided by Grail and Guardant, which collectively analyzed about 200,000 samples. With the vision of B2B model, initially Dogma Bio will target these major companies and subsequently hospitals and research centers, thus ultimately benefiting cancer patients. The number of cancer cases are expected to increase up to 28 million by 2040, and the liquid biopsy market will rise up to $5,000 million by 2027. Thus, by assuming that we acquire 50,000 samples in the first year of launch, and with 10 to 20% growth in subsequent years, our revenue can grow up to $2 million in 5 to 6 years. So far, our methods have been published in reputed journals. We have also filed an international patent based on these scientific innovations. 2 software solutions are production ready and can be used to demonstrate their value to potential customers. Currently, we are optimizing a new product, SeqWatcher, to target companies like Illumina for calibration of DNA sequencing instruments. This year, we will begin the design and deployment of website and cloud infrastructure. Marketing campaigns will be carried out simultaneously. Along these lines, we have registered our domain, dogmabio.com. In the long term, we have planned to develop an integrated solution with software and instrumentation support for early diagnosis. To achieve these goals, we are looking for $400,000 in funds to complete product development, cloud deployment, beta release of software, marketing campaigns, office setup, and to hire key staff. Our Dogma Bio team with 4 members has a collective experience of close to 40 years. Dr. Shautuma is an assistant member at St. Jude. With expertise in statistics, he will be serving as a scientific advisor. Dr. Pandurang Kolekar is a bioinformatics scientist and will undertake product management roles along with marketing. Dr. Corey Zhu is a senior bioinformatics scientist at St. Jude and will take care of project management, operations, and marketing. Ms. Nareen Mirza holds MS and MBA and currently pursuing PhD at University of Memphis. Her experience will be useful in marketing, finance, and operations. Thank you very much for your time. We would like to thank and acknowledge organizers for their guidance and constructive feedback. Thank you.
Thank you. Okay, we'll open it up to questions from the panel.
Thank you for that. Just making sure I understand what the product is. So the main part of your invention is some computational set of algorithms to reduce sequencing errors?
Correct.
So you are not solving the problem that you had kind of on the left of that slide, that there's very few cancerous cells maybe in the liquid biopsy. You're not amplifying that, or are you?
No, the existing methods, they require this UMI-based redundant sequencing to amplify the signals. but their sensitivity is low. Based on mathematical modeling, we have optimized our software so that they can correctly pick up the true mutations and suppress the sequencing errors so that we can increase the sensitivity of our method detections.
Is there any IP around this, or are you keeping it as a trade secret?
We have filed a patent recently.
So I guess I'm a little curious, this is kind of getting into the diagnostic area, which is a very challenging area in clinical medicine. You'll have, an initial advantage in the marketplace to start, but, as that information becomes more available, there may be others who can follow quickly behind you to do the same type of testing. Do you have any response to that?
Not as of now.
Do you think that's true? I mean, do you think others will follow, that you'll kind of be first out in terms of that type of sensitivity or variance, if you will, but maybe will become more commonplace after
sequencing? Okay, but compared to that, we have unique advantage that we do not require this redundant sequencing, and accordingly, we'll be saving the amount of data generated, storage, and turnaround time involved. So that way, we have unique advantage, and not just cancer diagnosis, but monitoring these mutations is also helpful in other industries like beverage industries, where they have to constantly monitor the mutations in their, microorganisms to ensure the quality of their products, and also on antibacterial resistance.
And I also want to, on top of that, I want to add that we're also developing other products such as indel and SV detection. So I think even like somebody catches up on the single mutation side, we're still holding advantage on other type of mutation detection. Right. So we have a series of pipeline going on.
And we are developing this product SeqWatcher. So every sequencing lab around the world, they need to calibrate their instruments. So using these products, they will help us to monitor and track the amount of errors in their sequencing instruments. So given the number of different products and number of algorithms that we have devised for sensitive detection, we'll be way ahead of the other competitors.
That kind of brings up a
Go ahead, Kayla.
Go ahead. Well, yeah, I think I'm curious about who is actually writing a check for this product when you bring it to market. Is it the hospital? Is it a sequencing company? Is it, who's actually paying for this bill?
The companies and hospitals who will be uploading the sequence data to us.
So yeah,
okay, so the hospitals write a check for this already, or is this something new that the hospital is going to have to do?
So are you talking about buying our service, writing the check to buy our service with general liquid biopsy service?
Yeah. So who's going to pay your bills? When you bring this product to the market and it's offered, where do the checks come from?
So
do they come from a hospital?
The check. So initially the check will come from the company. So our first target is the companies. If you go to that slide 12. Yeah, so our first target is Grill and Garden Health, companies like this. So we plan to, basically enhance their liquid biopsy accuracy. So we charge them $10 per sample. But as time goes on, we also plan to offer a similar service to medical centers or cancer centers. And eventually we want to, if patient they receive their liquid biopsy report from these companies or cancer centers, but they want a second opinion, they can reach out to us, and we would basically do the service for the patient for free. because the patient can advocate for us, because we basically provide a second opinion for them. And our method is actually have a higher sensitivity in terms of detecting low frequency mutation in the plasma.
Right.
So you have a whole range of products that you have listed. To get the most benefit, I guess, with your suite, do they have to adopt each of these? I mean, instrument calibration, all of these, I'm a little confused about how some of that works. And then along with that, is the equipment used so standard that, and the data output so similar that you don't have to develop algorithms for each type of equipment that this testing to be done on? Or is it one product suite that works with everything?
Yeah, so existing software is devised for measurement of accurate SNV, single type of substitution changes. But we are also developing these methods for other types of mutations like structural variants, which are long in the length as compared to single substitution changes, and also for indels. So ultimately, we will integrating all these methods into one single product, which will ultimately use to improve the accuracy of the report. And Sequatcher is another product which will be used for a separate purpose to monitor the accuracy of the sequencing instruments.
On the back end, not.
Yeah.
Okay. So they're just uploading reports to you strictly. You're not having to do anything on the front end side of their.
Right. They'll be uploading just data, and we'll be taking care of the reports and assessing the accuracy of the instruments.
I think that might be I get a little lost when you have all these products, but then maybe it's really just one service.
Yeah.
Just from a flow of the presentation, it might be easier to kind of collapse some of that.
Mm-hmm.
Okay. Team 1, great job. Great questions, great responses. Everybody give Team 1 a round of applause. All right. Team 4, please spool up your presentation. We'll get started when we see you in full screen. And don't let the screen fool you. This is not Ryan Hughes about to do this presentation. This is his better half, Jesse Hughes, making this presentation, bringing home Team 4. Okay, we'll give you a countdown, 3, 2, 1. Ready? 3, 2, 1, go.
Hello, my name is Jessica Hughes, and I am proud to present our company, MediSense, to you tonight. Imagine walking into the doctor's office because you had a headache, or you felt tired, or were experiencing weight loss, only to walk out being diagnosed with the unthinkable, cancer. Now, maybe some of you don't have to imagine. Maybe someone here has gone through this experience. Maybe someone you love has been through this. The feeling of pain and helplessness. I know I don't have to imagine. My Uncle Tony was diagnosed with pancreatic cancer back in 2013. Sadly, his disease progressed like so many others, rapidly and without any signs of remission. He passed just 6 short months after initially being diagnosed. Our family had so many questions. Why wasn't the cancer diagnosed earlier? Why weren't there any symptoms until it was too late? Could we have done something about it if we had known about it sooner? We'll never really know. And here's the scary thing. Over half the people living in the world today will be diagnosed with cancer at some point in their lifetime. That means half the people here tonight will come face to face with one of the world's deadliest diseases. This equates to more than 14 million deaths a year. With all of the emerging treatment options available, why is cancer still such a deadly problem? What really causes cancer deaths? And as it turns out, 90% of all cancer-related deaths are caused by metastasis. As you can see, survival rates once cancer reaches stage 4, when metastatic tumors are already established, are greatly diminished. If we can shift the timeline by identifying cancer before a patient begins to experience fully developed metastasis, we can save lives. Because it is so deadly, detecting metastasis early and monitoring its progress is critical for the health outcomes of patients diagnosed with cancer. The most prevalent methods for detecting metastasis involve multiple blood tests, time-consuming and expensive imaging tests, of which there are many, or just waiting until a patient presents with abnormal daily symptoms. And by that point, it is often too late. In recent years, research has shown that metastatic cancer has a hallmark: circulating tumor cells, also known as CTCs, in the blood. Cancer spreads through the blood via CTCs or clusters of CTCs, which then establish metastatic tumors in other areas of the body. Unfortunately, CTCs are rare. In 1 ml of blood, only a few CTCs are found compared to 10 million white blood cells and 5 billion red blood cells. Various CTC isolation and detective methods have been developed over the years, but these methods are technically costly, complicated, and insensitive, meaning patients may receive a clean bill of health when their body is actually battling the early stages of cancer. At Medisense, our technology integrates immunomagnetic isolation and detection by a common method known as SIRS into a single step using iron oxide gold core nanoparticles. These nanoparticles use multiple surface markers to bind directly to the CTCs in whole blood, trapping them for magnetic separation. SIRS simultaneously detects multiple targets, and the unique shape of our nanoparticles increases detection sensitivity even more. Our research shows that our system can capture up to 90% of CTCs in a blood sample. A simple blood draw is used to obtain the 1 ml sample, and no pretreatment is required. So this test could easily be performed at a patient's annual physical as a preventative measure. And since our assay's single step is completed in 2 hours, it is more efficient and less costly than current detection methods, and patients get their results sooner. As global rates of cancer rise, healthcare providers are using diagnostic screening tools more often and are constantly seeking better and earlier detection methods. Currently, the global cancer diagnostics market is a $41.5 billion industry. By 2027, this market is expected to grow to $83.6 billion, doubling in less than 10 years. Of this global market, tumor biomarker tests like ours are by far the largest and fastest growing segment. Unlike the typical lab and imaging tests often used to diagnose cancer, our approach is able to detect CTCs before a metastatic tumor forms, giving providers early insight into a patient's prognosis. Because we use multiple surface markers, we can detect multiple types of CTCs, meaning fewer false negatives for patients and making our technology applicable for more types of cancer detection. Not only is our process more sensitive, but our one-step approach to isolating and detecting CTCs is faster and more cost-effective than the competition. And since our test requires only 1 ml blood draw and no pre-treatment of the sample, it can easily be integrated into regular checkups with a primary care physician. Our intent is to license the technology to lab service providers like LabCorp, while also potentially entertaining licensing agreements with the largest cancer diagnostic test manufacturers like Roche and AstraZeneca. We plan to promote our technology by continuing to publish robust data, demonstrating the efficacy of our technology, and by attending conferences and trade shows to inform the medical and research communities about the advancements CTC detection we've made. In 2017, the patent for our technology was granted. Since then, we have also published our research in various journals, including Nanotheranostics and Applied Materials and Interfaces. We have begun preclinical trials using a breast cancer model, and in 2020, we were awarded an R15 grant for this research. In 2021, we participated in the Memphis Scipreneur program and had the opportunity to develop a business plan. Today, we are seeking a total of $150,000. About $35,000 will go toward continued research and developing a prototype. Another $37,000 will go toward registering our technology with the FDA, as a new 510(k) Class 2 device, as well as the associated legal consulting fees. And finally, most of that money will go toward continuing preclinical trials and expanding into other models for cancers that commonly metastasize, like prostate and colon cancer. We have a multidisciplinary team of founders with years of experience in scientific research, consisting of myself and my co-founder. And we've been greatly assisted by our wonderful mentor, Dr. Huang, who developed this technology with her colleagues at the University of Memphis. With your help, we will be one step closer to saving lives with our groundbreaking technology so that people like my Uncle Tony can have a fighting chance in beating cancer. Thank you.
Thank you very much. That was fantastic. Everybody, let's give them a quick round of applause before we go to the panel. That was a great pitch. All right. Let's kick it off. Lisa, I think you were going to start.
Yeah, Jessica, thank you for that. I just had a lot of questions because, you're kind of, I guess, overlapping into some of my, my company's activities and interests. You mentioned that you could detect this before metastatic tumor forms, and I'm just curious what data you have to really back up that claim. And so, I realize this is kind of an early detection, but do you have actual data that demonstrates that you can do that?
So we have not done that in a live mouse model yet. So this detection method is based on the research that they've done in mouse models to date, but we wouldn't actually have that data yet. That's what we need a little bit more time to research, but we definitely have models that have been done like in a Petri dish where from a very low cell count, they were able to detect the CTCs.
Okay, and then I have lots of questions, but my last question, you mentioned about a ml of blood, and what we do is, oftentimes we're asked to do specialty assays for clinical trials or even for patients, esoteric biomarkers, which this fits into. You talk about a ml of blood, but have you kind of walked through the process? Like once that ml of blood is obtained, from the patient, then what are the options? Because, we're often asked, can we ship this ml of blood to you? What's the stability of that sample? What's the turnaround time that you need? Maybe this is you're at an early stage to think about those, but that's very important when you think about giving a ml of blood and to be screening for this type of, say, early detection system. You have to be able to kind of think about what happens after that ml of blood is drawn. Can that be shipped? Does that need to be processed to a certain extent? What's the viability of those cells? And I don't know if you've had a chance to kind of think that through.
Yeah, that's a very important question. Obviously, we want to make sure that any samples that we're taking in are preserved in such a way that they're still useful and that they're providing accurate results. So for this actual assay to work, there is no pre-treatment of the blood that needs to happen. It is just whole blood that's run through contact with these nanoparticles, and then the nanoparticles are binding with the circulating tumor cells. So there's no actual treatment that needs to be done. But as far as keeping the whole blood safe, yes, that would definitely be necessary. And considering we're wanting to license this to LabCorp, we would be utilizing their resources and their systems for shipping. And as far as how long it takes to get results, our assay from start to finish is done in 2 hours, completely done. Whereas the currently FDA approved one takes a couple days to complete. So that would also increase the turnaround rate for results.
Yeah, and I mean, LabCorp is obviously a large company. I think in some ways you need to think about, maybe some other opportunities because, LabCorp is a huge company, but, in terms of specialty assays, you may have some other options that in the end might serve your pharma customers and your patients a little bit differently.
Great, thank you. It's a wonderful suggestion.
I had a business question about when you are successful. So first off, fantastic job. But when you're successful, you show those numbers up front about, stage 4, stage 1 detection. I'm curious if you've done some calculations on what this could save the healthcare system. Not necessarily the dollars of the diagnostics, but when we're talking about being able to detect something sooner or having that information, curious on what you've deduced from that.
I have not crunched those numbers. I do know that just baseline, our test would be quite a bit cheaper than what's currently available. That's our main competitor. It would probably be almost 10-fold cheaper. So there's that for the detection method. But we know that tumor biopsies on top of that, which would be a much more late term detection method, those cost thousands and thousands of dollars. So I think if this were rolled out in a significant context, you could expect there would be something in the order of millions that you would expect to save, because you're looking at over time the cost of chemotherapy treatment, the cost of checking on those tumors, removing those tumors, and then unfortunately end of life care. I mean, it's all going to add up.
Yeah, I would say it'd be, since this would be positioning it towards payers have to, they ultimately have to cover this. Being able to say that out of 10 patients, we're going to get one of them when no one else would have, and of that one, that cost would be X. Building that business case would be really powerful.
Thank you.
Yeah, I agree with you, Kayla. Unfortunately, the insurance companies don't care about patients for more than about 3 years. So looking 5 years ahead, they don't really care about. What cancers can you detect, and would you have to have a separate kind of binding mechanism to your nanoparticle for every type of cancer, and is that a separate FDA submission each time?
Yes, so you would have to expand the FDA approval each time you add a different detection method. Luckily, these nanoparticles, you basically just coat them with whatever antibody you want to use for detection. So far, our technology, we've tested it with HER2, in addition to EpCAM, which is the epithelial cell signaler antibody, so it captures both types of CTCs that would be in the blood. But we would, in getting FDA approval, have to expand on that approval process each time.
Thank you.
So with your technology, does it still require, I know like cell search, they say they're semi-automated. Do you still require a diagnosis by a pathologist or a cytopathologist? Or are you trying to more automate that system, I guess?
I think you would still, obviously, the healthcare provider would be the one interpreting the results of the test. But the way that the SIRS, the spectroscopy works, it actually releases a spectra. And at certain points along the x-axis, you know exactly what that spike is. And then on the side, you have the frequency or the intensity of that spike. So you can actually determine, okay, here for this antibody, we got a spike that's equivalent to 2 cells. And in the case of circulating tumor cells, that's quite a bit. That's a lot. So the spectra is actually very easy to understand and read. And I think it's very quantifiable. And you could just be able to share those quantities with the healthcare provider.
Okay. Team 4, Jessica, great job. Thank you very much. Another little round of applause for everybody. Great questions. Thank you, panel. All right, we're going to move on to Team 5. So Team 5, when you're ready and your presentation is in full view, we'll get you started.
Yeah, it's difficult to present after all this wonderful presentations.
You'll do great.
You got it.
Yeah, they've all been fantastic. No pressure. Okay, I'm going to give you the counts and then we'll get started. 3, 2, 1.
Hi, my name is Fatima Karamatnia. I'm a PhD candidate in the Pharmaceutical Sciences program at UTHSC, and I'm representing GBM Neurobio. Glioblastomas come back, sometimes sooner, sometimes later, but they always come back. That's the nature of GBM, says Bob Kemp, a 4-year survivor from Westchester, Ohio. Bob is a 62-year-old trim and athletic man who participates in 5K for a brain tumor cure every year, but he is living with the odds. The average lifespan of a GBM patient after diagnosis is 12 to 15 months. This is the most invasive type of brain tumor and accounts for more than 50% of all primary brain cancers. Treatment is started with surgery to remove the tumor as much as possible. Then patient goes to cycles of radiation therapy and chemotherapy. But as Bob says, glioblastomas come back, and in fact, in 80% of patients, relapse happens. This standard of care not only has low efficacy, but it's also highly toxic, leaving patients and their caregivers helpless with poor quality of life during this short lifespan. What is more frustrating is that during the past 3 decades, GBM treatments have only improved the median survival of patients by an average of only 3 months. This devastating disease has a global impact, with high national burden in the United States to Europe all the way to Asia, and every year, 3 new cases per 100,000 individuals are added to this population of GBM patients. Normally, male individuals with the average age of 45 to 70 years old are at higher risk for GBM. However, family history, genetic background, and some environmental factors play a critical role in causing this disease. The median age at diagnosis is 58, and the median age at death is only 7 years later at 65. To put this in another perspective, imagine only 35% of patients survive during the first year of their disease, and this number shrinks to less than 5% after 5 years of diagnosis. The first-line therapy for GBM is chemotherapy using temozolomide. This chemotherapy is able to destroy the fast-growing cancer cells. However, cancer is smart. They have a small fraction of stem cells or glioblastoma initiating cells, which are resistant to this therapy. And later on, they grow and they cause relapse of the GBM. Now that we know cancer is smart, we have to be smarter. With our novel bromodomain inhibitors, we are able to selectively target these tumor initiating cells. And once combined with the first-line therapy, we would be able to eradicate tumor at early stage and significantly improve the current standard of care for GBM. While current treatments require high doses of chemotherapy in intense cycles, we believe that using our combination therapy, we would be able to improve the effect of first-line therapy and consequently reduce the toxicity and quality of life of the patients. On the other hand, considering the current survival rate being 35%, our initial studies in GBM mice models support that our combination therapy has the potential to prolong the patient's survival and, most importantly, decrease the rate of recurrence, which happens in 80% of patients. There are few therapeutic options to battle this deadly disease, and our combination therapy using novel bromodomain inhibitors meets all the requirements for a safe, effective, and feasible treatment strategy, while our competitors suffer from lack of efficacy, toxicity, and complications of development or administration. It takes an average of 10 to 14 years, more than $600 million to identify a drug for a specific target until it reaches to the patient's bedside in the clinic. It starts with basic research, preclinical development. The promising result will lead to IND application, where FDA will approve testing the drug in human subjects in different clinical trials. And if the benefits of the drug outweighs toxicity, it will get FDA approved and reach the market. Here's our progress. In 2018, we identified these novel bromodomain inhibitors and their mechanism of action. During the past 3 years, we've been able to complete proof of concept studies. We have filed international and US patent. We have granted some institutional funding, and we are in the process of submitting a grant application to NIH to get funding for our next step, which is establishing preclinical studies. We are planning to attend conferences to spread the word and have early conversations with healthcare stakeholders, and also partner with biotechs and big pharma who have established pipeline for GBM drug development. And currently our treatment falls in the largest segment of global share market for GBM by treatment, and the projected revenue is going to be over $7 billion by 2030. We know that we are still in the preliminary stage of this long and costly process, but we believe that our treatment has the potential to become a viable candidate for FDA approval. What we are asking here is $500,000 to establish our preclinical development, which will provide for our efficacy, toxicity, and analytical studies. Our team consists of scientific professionals, Dr. Pfeffer, who is a professor in College of Medicine with more than 3 decades of experience in cancer research, Christina Wang, and myself. We are young but motivated and creative PhD students. We also have the support of a great team of advisors. And at the end, we believe that scientists, clinicians, and business people, if they work together hand in hand, they could give GBM patients and their caregivers, people like Bob and Janice, more time, more chance, more hope to live the quality of life they truly deserve. Why not us? Why not today to take the first step to make this dream come true? Thank you.
All right, great job, Team 5. All right, we're going to open up to the panels.
So for a non-scientist, which I am, is the main thing that you do is eliminate those stem cells, and that's
Yes.
why the reoccurrence is less, et cetera, et cetera. So the rest of the benefits are kind of downstream from eliminating those tumor stem cells.
Yes, that's true.
I was curious from an IP standpoint, based on Jan's comment. So what does your IP involve? Is it that elimination? Is it a change? You know, so the patents or patents pending, what does it help protect for you?
The patent is the compounds, these novel inhibitor compounds that have a new mechanism of action, which enables to make the cells more sensitive to temozolomide, the first line therapy. So these are actually chemical compounds, yes.
Got it. Have you heard of breakthrough status at the FDA? For 510K, or actually you'd be a PMA, but there's some really interesting things as you're writing these grants to just what FDA and Center for Medicare and Medicaid are doing with breakthrough. It's now, it's in the works. I don't think it's been passed, but it's likely to be passed that you get reimbursement if you are a breakthrough product. And what's really nice is that will significantly accelerate the commercial path, which would be highly beneficial for whoever you license this to or sell it to. I just think you have to do it very on and if you write that, I think in the grants as part of your regulatory path, I think that could be really helpful.
Okay, that's wonderful. No, I didn't have any information regarding that. Thank you.
So I'm just a little curious, what is exactly your target?
It's called RBG1. It is a part of a chromatin remodeling complex, which makes cells, resistant to DNA damage and temozolomide's mechanism of action.
Can I open my mouth on this? Since I'm the
Yes,
scientist?
yes.
So we identified that this specific part of the complex controls the DNA damage response. And we made drugs that specifically target one part of this drug. And we've shown that it binds to it specifically, doesn't bind to other things, and it's very tight binder and isn't a new kind of drug.
Mm-hmm. Okay.
Is this limited to glioblastoma, or is it in general for other stem cells?
I thought Dr. Pfeffer is going to comment on that. I think it has the potential to be used for other glioblastomas too, based on my understanding. Is that true? Right.
It can be used for other cancers. This is an unusual protein because it's usually mutated in a lot of cancers, but we found in a certain subset of cancers, it's highly overexpressed and can be targeted. And based on some of the comments earlier, one of the diseases in cancer, pancreatic cancer, it's highly overexpressed. We just haven't studied it in other forms of cancer yet.
Mm-hmm.
Other questions?
No, I mean, in general, it's obviously very early days for this one. I think it has super high potential. I think definitely look at maybe the other diseases. I have no idea how much GBM is versus pancreatic cancer versus others. But obviously, you'll get more excitement from investors if you're targeting a very much occurring cancer that doesn't have a lot of good treatments. So definitely let the market guide whether your next step is maybe looking at another cancer versus this one, or whether GBM is the first one you want to take to humans.
Mm-hmm.
Okay, that was great. Thanks again, Team 5 did a great job. One final round of quiet applause and emojis before we move on to the next step. Before we do, I just wanted to thank all the teams and all the participants from this entire program. You gave it your all. You showed up every week. You showed up to our office hours. You showed up to our special pitch practices. You made time with our guest speakers. It's been a fantastic experience. Y'all, I just wanted to draw this, draw attention to the team cohort here, all the teams together. They've done a fantastic job. So another round of applause for the entire group. Okay, so what we're going to do now is we're going to break out into breakout rooms. We're going to have one room with the judges and the coordinators, and they're going to deliberate for a little while. They've been keeping score in real time, so we know we can get this done inside of 10 minutes. The rest of y'all are going to be broken up into another half a dozen breakout rooms. You've been randomly chosen just in each one of those rooms. You can socialize during that 10 minutes while we're deliberating, or like I said earlier, take a bio break, grab a sandwich, rally the kids, do whatever you want to do. We're going to get started and be back in about 10 minutes. So I'm going to do that right now. Hey, Ryan, do you see the ability to join? You're on mute, by the way.
Hey, Brian.
Yeah.
I'm trying to swap computers because the computer I'm on is going to room 6, not room 1. So
yeah, I'm not sure. So does that mean Jessica's in the room?
Yeah, we're switching. No, she hasn't joined yet. So we're switching computers.
Okay. All right. So they've already started. Yeah, perfect. That worked out great, you two. Thank you very much. Because technically I'm trying to figure out how to do what you two did physically. All right. See you in a bit.
I think I have a tie, so I think I can, I don't know. I mean, I'm very open to just a conversation too of who everyone maybe thought was best. I don't know.
Y'all can talk away at it while I look, but also if it does come down to it being really close or a tie, then y'all can decide if you want it, for sure.
Yeah.
Just catching up here, is there not a clear winner? We have two that are.
I'm still going through to look.
Yeah, we kind of have to, should have made a fifth sheet that adds them up. Team 3, team 2.
Oh yeah, and I was so mine are, I wasn't sure what the teams are, so I put the names in, which made it probably much worse. So would you mind just telling me maybe really quickly who the teams are?
Yeah, go ahead, Shelby. Do you know?
Can you look at that, Brian, while I'm sitting here and counting through these?
They need to put that on their presentations in the corner next time.
Yes.
Yeah. Or if you guys know the order, you can just put them in here too.
Yeah, so we had team 6 first. That was Tummy Time. I'm drawing a blank as to what the name of the company for the second one is. Team 3 was the Kaizen patch.
Protocure.
Oh, Protocure. Right.
With team what?
That was team 2, which came, this is the order that they presented in. So team 6 was Rohan with Tummy Time. Team 2 was Protocure. Team 3 was the Kaizen bandage. Team 1 was the sequencer, the software. Team 4, Jesse, was the first of the 2 cancer cures and then treatments, and then team 5 was the GBM.
So team 1 was Dogma Bio. Dogma
Right? Bio, right? Yes, Dogma Bio.
4 is Medicines.
Madison's.
Yeah, Shelby, mine, I'm happy to reorder mine if it's helpful into 1, 2, 3. I have the names and then the teams up top, but I don't know.
I've got it.
I can tell you my favorite teams. Yeah.
So you guys, it's tied right now between Medicines and Protocure. Essentially.
Those are my top 2.
Yeah.
Medicine and Protocure, which was not team 2.
Protocure was team 2. Medicines was team 4.
Yeah.
And thanks for it. We'll make that clearer next time. So appreciate that.
Yeah, I mean, my issue with team 2 is that I have no clue what it is. I mean, it was 10 slides of science. Might work, might not work. I just don't know. And I thought Medicines, which was my top team, I thought it was just a really good pitch. I mean, it kind of touched on all the things, didn't get too deep into science. I mean, there's still plenty of open questions, but I thought overall was the one that impressed me the most, being the most complete.
I completely agree. That had like a beginning, middle, end. She tied the story together, and that was not just the science, but that was a business pitch too, which is the goal for me of this program. Is you've got to translate the science into something digestible that an investor can understand. And I think pulling on the emotions is helpful and powerful, and having stories that resonate that you kind of wrap up at the end. So I was extremely impressed. It was like a breath of fresh air when we got to like that.
Yeah, especially when you have such far out, like long-term ideas. So it's hard to really parse through and say which one's gonna, inherently be better than the other. So I think,
I'm positive about Medicines. I think the thing that kind of gave me a little pause was that you really can't claim, that you're able to detect these cells before metastasis. That's a huge leap in terms of the technology until you prove that. And then just, what the mechanics are for the samples to be actually analyzed from the clinic. So, that's kind of a little bit near and dear to, what we do on a daily basis. So, making those claims before you have data, is a risky type of thing. I like the concept. So, I'm not certainly, they got a good score, for me, but those were two things that were a little, maybe I didn't give as many points for that. And I think the reason that I gave Protocure more points is that I really do understand the science behind it, and JAK2 targeting is really the hot topic in terms of immuno-oncology, and having something that, may target something a little alternative to the JAK2, but, affects JAK2, was intriguing to me. And I thought, maybe a novel approach. So I know we need to look at it from a full, presentation standpoint, and, what was compelling to the judges as a whole, but that's kind of why my scoring was, what it was.
And I really liked Protocure as well. I thought of the scientific presentations, especially the significantly scientific ones. I thought he did amazing on it. I felt like I could follow it, which was really rare for me. So I very much appreciated that. And I thought that, I don't know, the visuals were really nice and not overly complicated. And I think that's very important. They were tied for me with medicine.
He was in that lab, I assume. Is that?
Had to be.
Like, I'm sure he was, yeah.
He's bioinformatics.
I feel like
maybe.
Well, it sounds like we have a first and second. Do we have a third?
My third was GBM, so team 5.
I picked that one. I knew it was going to be 2 or 3.
They had the best looking presentation for sure.
So is that agreement from the panelists for MetaSense 1, Protocure 2, and GBM 3? Your call.
How does that jive with the scores? Just out of curiosity.
It actually looks like it would be between GBM or Chitosan for third.
Oh, but the other two were first and second.
I would say, based on y'all's feedback as well, I would say MetaSense was first. I think Josh and Kayla, you guys had ties between them. So feedback wise, MetaSense wins, Protocure gets second, but GBM and Chitosan are technically tied for third.
I'd say GBM.
Okay.
I have a hard time with Chitosan because I'm too close to it. So
fair enough.
So if we dismiss your low score, they might be bumping up higher, but
I agree.
think GBM,
I mean, I wonder if I'm.
Yeah, to me, GBM just has a lot of potential. If you truly can knock out these cancer stem cells, and I'm not sure GBM is the first indication they should go after. But I mean, it's the reoccurrence is what kills so many patients, right? And if they have some sort of platform to go after those cells, I think it's going to be huge in cancer treatment.
And I would say, just from a presentation standpoint, I feel like I was impressed by GBM. I thought they did such a great job answering questions versus I feel like in Chitosan, we got asked a lot of questions. Yeah. And so just from a pitch standpoint and an etiquette standpoint of the situation, I do feel like they handled it very professionally and really eloquently, which is why, I really like TBM.
Agreed.
Yeah,
and we're
Brad
only
is-
laughing because Brad is a question asker. That's all the only reason we're laughing. He's very
Yeah.
inquisitive all the time.
I've
Not
been
surprised.
I've been in Memphis for
It's a good thing.
7 years, and I've heard Kaizen every month for 7 years. I think.
Yeah, I think, it's a usual
irritated by that.
suspect.
It's good stuff. It's good
Yeah.
stuff.
Yeah, no,
I believe in it.
I coached a team last year on the same thing with the same tech, same leader, everything. So, all right, I think this is great. We need to get back to the other rooms. So if we have consensus on 1, 2, 3 being Medisense, Protocure, and GBM in that order, I think we can go with that. And Erica, that's going to be on you to announce those when we come back, or after we let Abby go, and then we'll announce the winners.
On the inside joke, you're going to have to announce Erica's Dr. Dillard. Dr. Dillard.
Yes.
Medicines, Protocure, and GBM.
Right.
Yes.
Announce them in reverse order.
Say what?
Announce them in reverse order.
No.
Yeah, go.
Don't mess me up, Ryan.
No,
Do you wanna go
yeah. Third
In
place will be
third
GBM.
place.
Announce that first. Then
Okay,
Protocure,
third
which will be
place,
second.
GBM. Okay, I'll do that.
Yeah.
I got to
That's
switch
great.
your room, but I got
All
it.
right, here we go. We're gonna go all back to
got
the
this.
main room. Thanks, y'all. Okay, everybody's starting to come back into the main room here. They do have to notice a little thing to switch it back in. So we'll just give another 45 seconds or so to let everybody in. "25 seconds, and then it's automatically going to dump everybody back in. So we'll just give it that period of time. The conversation in the deliberation room with the panelists was tough. There's a lot of great comments made. Everyone did a fantastic job. So it was really rather difficult to, pick those top winners. But in every competition, there has to be winners. But there are no losers here tonight. I think all of the teams did fantastically well, and there's a lot of opportunity for the technologies that they're working on to carry on past this program. And that is the goal of what we're doing here, is not just to run through a cohort over 9 weeks and then see you later and it's one and done. We really want to see these technologies transfer to the open market, and these are viable teams and solutions that have been brought to bear and presented tonight to have a plan to do that. So, it was really great to see. Okay, so next we're going to listen and hear from Abby Trotter, who's from the Bio10 group, and Abby's joining us tonight. And let's see. Hopefully,
I'm here.
Okay, perfect.
Yeah.
I still
Thank
see
you
some
so
people
much,
over there.
Brian.
Go ahead.
And congratulations to all of the teams for some great pitches and some really great technologies tonight. And I'll only be a minute because I know I'm standing in between you and finding out who won. Just a couple of things that I want to point out about Bio10. Number one, I want to recognize Stephen King, who you might see on your screen tonight. He is our brand new director of the Life Science Mentor Network, which is a terrific program that all of you should consider now that you've been through this process. If you want to take a piece of technology for real out into the commercialization space and get it ready for pitching, the Life Science Mentor Network is exactly how you do that. And you can go on the Bio10 website and get some more information. But Stephen has done work in Memphis. Many of you might be very familiar with him. He has been involved in several companies in the Innova portfolio, so has a wealth of experience with the Life Science Mentor Network. So I just want to shout out to him for being here tonight, and message him if you're interested in learning more about that program or get on our website. I am so thrilled to see the progress of the Academic Alliance and what the Academic Alliance has done under the leadership of Erica Dillard and this program and what Ryan has been able to do, and then pulling in CBU as well this year. Every year this program just keeps getting better and better. You know, just to give everybody just a short bit of history, the Academic Alliance predated me and my joining into Life Science Tennessee and what is now Biotin. It was started, I think, in the late 1990s or early 2000s actually. And in 2013, Chad Riggs reminded me that Cliff Herwick actually started the program in Memphis. And then in 2015, the predecessor to Scipreneur, the Tech Venture Challenge, got started at the Nashville chapter of the Academic Alliance, or the Vanderbilt chapter of the Academic Alliance. And then Memphis, as Memphis always does, raised the bar and changed the name to Scipreneur, made it that much more marketable, and started that program in 2017. So it's terrific to see 5 years of the Scipreneur program. In 2019, the Knoxville chapter of the University of Tennessee at Knoxville chapter of the Academic Alliance got started, and they actually did their first Scipreneur program. So there's a long history of great programming with the Academic Alliance, and particularly the Scipreneur event. And then just to give all of you a plug, the job fair coming up on May 1st is going to be another fabulous event. The first one that we had in August of last year was great. We've got more companies coming on board. We've got more job seekers that are going to be there. Look, you can check it out on our website as well. If you can't do just the job fair, also think about the resume workshop. If you just want to hone your resume a bit for when you're ready for the career, for the industry, you can do that by going on the resume workshop, going on our website, signing up for the resume workshop. So make sure you take a look at all of the Biotin activities. We're doing great things. Take advantage of all of this. Make sure you get our newsletter. If you don't, let me know, shoot me a note. You should get the it has a lot of logos and pictures all over it. It comes out about once a month. So make sure you get the Biotin newsletter as well. I know many of you also get information from Deepka. So, but thank you so much, Brian. Thank you to CBU for elevating all of this. Thank you, Ryan, for all of your hard work. And appreciate everyone being involved, and good luck to all of you. I'm anxious to hear who the winner is.
Thanks, Abby. Erica?
All right, the moment has come. Let's see. All right. What's going on? Can you guys see my screen?
Yeah, beers is all I see, though.
Beers and careers, that's right. All right. Well, we'll start with third place winner. For third place, we have GBM NeoBio. Awesome job, guys. Awesome job, Christina and Fateme and Dr. Pfeffer, for a wonderful job this whole time. You guys had a wonderful, a great presentation, and it's been a pleasure working with you all. So pat yourself on the back. Any judges want to say anything, like a statement at all about team 3 or third place? Maybe not. All right, I'll keep going. All right, and so for our second place, we have Proteolytic. Pankaj and Caitlin, awesome job, Dr. J.K. We really appreciated this. Really strong on the science, and we definitely look forward to what you guys are going to do in the future. And last but not least, we have Medicine. Jessica killed it. Really appreciate this presentation, and Dr. Wong, for allowing you to utilize this IP. So with that being said, we will have your prizes for you. Everyone who is on each of these teams will receive a monetary prize, as we mentioned before. But everyone is a winner here. All the teams and participants will receive a gift of our appreciation for your hard work. We have a swag bag to give to everyone as well, including t-shirts for Memphis Scipreneur Challenge for 2021. We have some mugs and pins and some cool swag from CBU as well. So thank you all. Also, we have some gift certificates too, some gift cards for you guys to take advantage of. So congratulations, everyone. Really appreciate the hard work. And looking forward to having you guys present, as Abby mentioned before, having you guys present in November at the Life Science Conference. So great job, guys.
Yeah, great job, everybody. Congratulations to the winning team. Thank you, everyone, for joining us tonight. Panel, fantastic. Everyone look at Jan's little background thing. It's going crazy. He really pushes the envelope in Zoom, as well as buys into companies. So anyway, again, thank you very much for your time. It concludes the 2021 Memphis Scipreneur Challenge cohort. We at CBU really look forward to working with y'all more in the future. We really enjoyed working with Erica and Ryan. Shelby and I both did, and the rest of your team. It's been fantastic. Y'all have all done a great job. Very, very proud, very impressed. With that, I'm going to conclude the session, and we're going to say bye. Everyone wave bye. Everyone else, it was a great time, and we'll do this again. Thank you very much. Take care, everybody.